Hello everyone, and welcome to this audio. On in and around an overview in context of this research poster that has been put forward for the International Paediatric Transplant Association, or IPTA 2023. This works is an overview in and around transplant neuroimmunobiology, the immune system, transplant rejection, and neuroendocrinology, and it's a sort of a retrospective. And this was collaborative between several academics and academic institutions, and so here I'm going to provide a sort of overview around this research, providing beginning with the aims around this works. So the aims of this works are to highlight how specific immunobiological and neuroendocrine markers can reduce chemokine migration and acute allograft rejection, or AAR. And in and around the mechanisms, the pre-transplant phase in context of solid organ transplantation is just as central as the post-transplant duration, and it is postulated that Hans-Selye's general adaptation syndrome 1936 and specific neuroimmunobiology and neuroendocrinology should be considered aligned to transplant aligned to transplant immunobiology in context of allograft prognoses. And so, in and around the background around this works, cytokines. Cytokines are small molecules that signal between cells, inducing growth, differentiation, chemotaxis, activation, enhanced cytotoxicity, and/or regulation of immunity. Chemokines and cytokines which direct cell migration and/or activate chemokines are cytokines which direct cell migration and/or activate cells. Chemokines closely related to cytokines are produced by many cell types in response to the infection or physical damage. Chemokines activate and direct effector cells to sites of tissue damage and regulate leukocyte migration or trafficking into tissues. In context of immunobiology, CC chemokines are chemotactic for monocytes, and CXC chemokines are chemotactic for polymorphonuclear neutrophils, or referred to or abbreviated as PMNS. And chemokine receptors are expressed on specific cell populations, permitting different chemokines to have selective immunobiological activity. Chemokines are primarily involved in attraction of leukocytes, so for example, lymphocytes, monocytes, and neutrophils, and they can be released by many different cell types and serve to guide cells involved in innate immunity and lymphocytes in adaptive immunity. Some chemokines also have roles in the development of lymphocytes migration and angiogenesis, and where angiogenesis is the growth of new blood vessels. And it is evident that cytokine molecules are important molecules for activating and directing effector cell populations to the site of damage, as well as for controlling leukocyte migration into tissues. So to attain long-term transplant prognosis or survival post-transplantation has been the ultimate challenge for a variety of organ transplant specialties. Immunosuppression, or IST, reduces the activation or efficacy of the immune system. Some portions of the immune system itself have immunosuppressive effects on other parts of the immune system, but clinically induced immunosuppression is carried out through pharmacotherapy, and this is usually done to prevent the body from rejecting an organ transplant or from the treatment of autoimmune diseases. And so when an organ is transplanted, the immune system of a recipient will recognize it as foreign tissue and attack it. And so cortisone was one of the first immunosuppressants identified, and the more effective immunosuppression, or IST, has been known as azathioprine, was identified in 1950, in the late 1950s, specifically 1959. But it was not until the discovery of cyclosporin, or cyclosporin A, in the 1970s, that transplant surgery found a sufficiently powerful immunosuppressive agent, or IST. Monoclonal antibodies at the same time, or MABs, are antibodies that are identical to the immune system because they are produced by one type of immune cell and all clones of a single parent cell. Monoclonal antibodies, or MABs, have advanced from involving fusion of immortal cell, for example, a myeloma tumor cell, with a predetermined antibody producing B cell, and this sort of fusion is done by ensuring cell membranes that are more permeable. Initially, mouse monoclonal antibodies, or MABs, did not make ideal therapeutics because differences between human and murine genetics. This has been dealt by humanizing murine antibodies, or by making fully human monoclonal antibodies. And so it has now become monoclonal antibody implementation has now become a sort of standard alongside mainstay IST. So the next part of this this works is to help sort of answer the question: how does neuroimmunobiology and neuroendocrinology associate? So stress is a medical term, or terminology for a broad range of strong external stimuli, both physiological and psychological, which can cause physiological response, and which has been referred to, where the physiological response, or even immunological response, has been referred to retrospectively as the general adaptation syndrome, first described in 1936 by endocrinologist Hans Selye in the journal Nature. Selye described the general adaptation syndrome as having three particular phases. Number one, alarm reaction, where the body detects external stimuli. Number two, adaptation, where the body interacts defensive countermeasures against the stressor. And three, exhaustion, where physiologically the body runs out of defenses. And so it would seem acceptable that stage two, or adaptation, where the body interacts defensive countermeasures against the stressor, corresponds to episodes of acute allograft rejection, or AAR. Now, studies of the immune system and psychology are becoming a direction which are significant and clinically important. A change in the amount of natural killer cells, or NK cells, and their stress-induced activity is an important defense mechanism in context of physiology, and it constitutes a component of the preparation for defense against potential pathogen penetration. So, for example, when people get angry, when individuals get angry, the entire body responds. It instantaneously becomes tense, and physiologically or automatically, the body moves towards fight or flight. And so when content, then physiologically the entire body becomes content. And stress hormones can change the immune cell behavior and the activity of the entire immune system. And investigations involving transplant immunobiology and neuroendocrinology should be included in investigations to better understand acute allograft rejection, or AAR. And this works has sort of a summary a summary sort of illustration highlighting the biology of chemokines and receptors. And so, for example, chemokines are involved in the development of T helper cell type 1, T helper cell type 2, angiogenesis, angiostasis, metastases, cell recruitment, inflammation, lymphoid organ development, and lymphoid trafficking, and also wound healing. The illustration has been adapted from works chemokine and receptors are as being important in a variety of immunological functions, and the illustration was adapted from works by Rossi and Zlotnick from 2000. Now, in context of this works as well, as it leads into providing a basis around the literature as a sort of a retrospective. O'Leary in 1990 highlight that chronic stress has been associated with suppression of the immune function, and there is evidence that the immune system may not adapt over time. There's works by Adder and Cohen in 1993 that highlight both conditioning and stressful stimulation exert biological effects, or biologically affects that can alter development and or progression of immunologically mediated pathophysiological processes. It was works by Dabar and team in 1995 that informed that significant and selective changes in numbers and percentages are identified in peripheral blood leukocyte during stress in subpopulations of rat models. It was works by Evans and team in 1995 that examined how stressful events and depression may affect key cellular immunological parameters in subjects with and without human immunodeficiency virus, or HIV, infection, and cytotoxic T lymphocyte phenotypes. It was Mills and team in 1996 that examined relationships between psychological characteristics and immune responses to an acute laboratory stressor, and such distribution may significantly affect the ability of the immune system to respond to potential or ongoing immune challenge. It was Shedlowski and Schmidt in 1996 that discussed stress on and the immune system, and acute stressors, and physical activity have been shown to transiently enhance immune responses. These stress effects on the immune system seem to be seem to be mediated through endocrine factors, since hormones, neurotransmitters, and neuropeptides can interact with cellular components of the immune system. It was Goebel and Mills in 2000 who examined the effects of acute psychological stress and exhaustive exercise on the expression and density of adhesion molecules. For example, L-selectin LFA type 1 and ICAM type 1 on monocytes, granulocytes, and lymphocytes. It was Segreger and colleagues in 2000 who highlighted chemokine receptors are present in acute allograft rejection, or AAR, and correlate with the distribution of T cells. It was Hancock and team in 2002 that highlight varied chemokine receptor expression in cardiac allograft rejection. It was Cockwell and team in 2002 who demonstrated the importance and the role of chemokines and chemokine receptors in acute allograft rejection. It was Larson and team in 2001 who informed acute stress may signal increase in at least some aspects of cell-mediated or T helper cell type 1 driven immune response. It was Maddock and Pariente in 2000 and 2001 who also say that chronic stress appears to result in suppression of the immune response, whereas immune activation and suppression have been associated with acute stress. It was Sternberg in 2001 who informed that physiological levels of glucocorticoids are immunomodulatory rather than immunosuppressive, causing a shift of cytokine production from primarily pro-inflammatory to anti-inflammatory pattern. And such patterns of cytokine production can be categorized as T helper cell type 1 or T helper cell type 2, and roughly correspond to cellular or humoral immune responses. It was works by Redwin and colleagues in 2003 who hypothesized that activation of psychological stress response increases autonomic activity and enhances immune function by inducing a significant increase in numbers of leukocytes at sites of inflammation. It was also Redwin and team in 2003 who further discussed that few human investigations have examined the effects of stress and leukocyte motility, and to date there are no published works examining psychological stress and leukocyte chemotaxis in this context. And chemotaxis did not correlate with catecholamine levels, suggesting that another mechanism of activation may also exist, which increases chemotaxis after stress. And it was works by Yun and team in 2004 who demonstrate that chemokine receptors play significant roles in the development of chronic rejection and may serve as potential therapeutic targets for cardiac allograft vasculopathy. And it was Xiaofeng and team in 2005 that demonstrate the unique role of chemokine molecules in the maintenance of cardiac allograft tolerance mediated by natural killer T cells in a mouse model. Ulig and Carlos in 2005 who informed that there are plurality that there are plurality or there is plurality of how endocrinological factors can affect or may influence immunity and the immune system mediated disease. And this is consistent and there are consistent and convincing reports of links between stress and disease onset and progression. Prospectively, the perfect model would be where patients are sort of stress free before transplantation, i.e. i.e. for example, i.e. wherein patients are on forms of replacement therapies and prophylaxis, and throughout time awaiting a transplant. This may well suppress those chemokine receptors that lead to acute allograft rejection episodes post-transplant. So, in context of this sort of overview, there is works in this overview that highlight chemokines and receptors identified in human renal acute allograft rejection, and again listeners are welcome to access this research poster presentation through the Renal Patient Support Group, the RPSG ResearchGate page, which and once sort of accessing this sort of table overview that looks at chemokines and receptors identified in human renal allograft acute allograft rejection, and there are the list of chemokines, the findings, the main sort of findings around laboratory based investigations, and then the reference alongside that as well. And so the works the table can be accessed from a paper that was that's referred to as chemokines and receptors identified in human renal AAR or acute allograft rejection, and it's works that was this table was adapted from works by Inston and Cockwell in 2002. In context of sort of prospective investigations, chemotaxis is methodology to investigate transport or migration towards a concentration gradient of chemoattractive for leukocytes or neutrophils. Leukocytes, so for example, neutrophils, monocytes, and lymphocytes, or other migratory cells. An upper chamber containing a suspension of cells is separated by a membrane from a lower chamber containing a medium with chemoattractant. Chemotaxis of the cells from the upper chamber into a lower chamber occurs over a predetermined duration. The cells of the bottom chamber may be counted directly, and so for example when using a homogeneous population of cells, or maybe stained for defined markers when assaying a mixed population of cells and analyzed through flow cytometry. Now, in context of using a T cell culture system in isolation, this would be helpful to observe chemotaxis as follows. So, for example, to observe chemotaxis for chemokine receptors, cellular adhesion molecules, and monoclonal antibodies using flow cytometry analysis to identify movement of particular cell types. and measuring stress neuroendocrine levels pre and post transplant would be complementary investigations. Monitoring stress levels in pre-transplant duration is and could be of could be and is of importance. A study with chemotaxis assays could potentially observe catecholamines with repeated stress, and this would potentially allow to help identify which hormones influence chemokine receptor migration. A study that incorporates chemotaxis and monoclonal antibodies with neuroendocrines post-transplant would also be complementary investigations. Comparing the pre-transplant phase and observing chemokine receptor and neuroendocrine with monoclonal antibody therapy would be important. And with this, examining effects of acute stress and exhaustive physical activity on chemokine receptor migration would be complementary. Studies with natural killer cell activity and cortisol with chemokine receptors in pre and post transplant phases could also be important. Conducting investigations that relate acute chronic stress, cytokine activity, and depression may show significant correlations between the immune system and neuroimmunobiology. Measuring neuroendocrine function with hypothalamic adrenal axis or HPA and chemokine receptor migration in pre and post transplanted recipients would be important. And so, in and around the discussion of this works, particular states, So, for example, anger and frustration are classed as acute stress and will cause an activated immune response, but not the same as positive stress. So, for example, motivational stress. Positive stress or motivational stress and physical activity propagates endogenous beta endorphin increase. Other neuroendocrine need to be better understood. Works by Sternberg in 2001 informs that, for example, that glucocorticoids suppress cellular adhesion migration, macrophage activation, antigen presentation, T cell receptor expression, T lymphocyte activation, proliferation, differentiation, including cytotoxicity, and B cell function, including antibody production. Chemokines closely related to cytokines, so for example, known as subcytokines, are produced by many cell types in response to infection or damage. They activate direct cells to sites of tissue and regulate leukocyte migration or movement into tissues. To date, professionals of transplant disciplines observe chemokines and immunosuppression, or IST, in sole organ specialties, i.e., so for example, a nephrologist with sole-based studies and investigations, investigations in and around renal pathophysiology, and compare to investigations of other organ specialties. Previous investigations to understand acute allograft rejection and cytokines are commendable, and results have proved progressive. It was Inston in 2002, and works by Inston in 2002, who hypothesized that there is an alternative pathway that causes acute allograft rejection in patients following CD25 monoclonal antibody therapy, CD25 or monoclonal antibody therapy. And it was Cockwell and team in 2002 to 2003 also hypothesized that in human renal transplantation, there are alternative pathways that promote the development of trafficking phenotype of T cells in patients that have received anti-CD25 monoclonal antibody therapy. Cockwell and team in 2002-2003 implemented and used a T cell culture system to analyze the effects of anti-CD25 on the expression of chemokine receptors CXCR3 and other significant chemokine receptors on the surface of T cells. Currently, no research links neuroimmunobiology and neuroendocrinology in context of transplant immunobiology from a laboratory perspective, and so there is a potential perspective for prospective efforts in these areas, but there would require a sort of multidisciplinary research efforts to acknowledge scope. And whilst there are complexities regarding transplant allografts whilst there are complexities when regarding prolonging transplant allografts in published efforts surrounding in published research research efforts surrounding the work this work. So one of the questions one additional sort of question has been put forward and thus would it not seem sensible to acknowledge that stress can cause a more activated immune response, and it is apparent that chronic stress and depression are linked and cause suppression of the immune system, whereas acute stress can potentially cause a lymphocyte proliferation and initiate chemokine receptor migration or trafficking to transplanted or organ and sort of instigate an acute allograft rejection episode. However, at the same time, the link between stress and transplant rejection is uncertain. The proposal of using chemotaxis methodologies would be an admirable opening point to and linking monoclonal antibodies with chemokine receptor migration and stress and cellular adhesion molecules to identify how stress could potentially propagate acute allograft rejection. And it would seem sensible to state that stressed post-transplant stressed post-transplant population will have an increase in cytokine production, and this could immunologically devastate allograft post-transplant period, or another pathway or cause another pathway for acute allograft rejection. In and around the sort of conclusions of this works, around this sort of overview, more work is required linking immunosuppression, monoclonal antibody therapy, and cell surface targets, and neuroimmunobiology and neuroendocrinology. The immune system has a suppressed action post-transplant, i.e. to reduce leukocyte proliferation, which would normally act against a stressor. So the stressor in this case, in context of an organ transplant. Now, the aims of this work were to establish and put forward an overview surrounding organ transplantation, neuroimmunobiology, and neuroendocrinology in context of transplant immunobiology from a laboratory perspective. And whilst literature informs that chemokines surrounding transplantation provide a basis, it is now anticipated that this work could be used as a guide to prompt procedures for perspective across that this work could be used as a guide to prompt procedures for prospective investigations across disciplines or specialties, and using a multidisciplinary research effort should enable professionals to come together to achieve a better understanding of transplant tolerance. Based on Selly's work in 1936 and the general adaptation syndrome and stage 2, where the body engages in defensive countermeasures against the stressor, it is proposed that chemokines appear to be integral players in this complex and dynamic process. A combination of neuroimmunobiology and neuroendocrinology in context of transplant immunobiology from a laboratory perspective to understand transplant immunobiology in context of stress. And Selly's work is important. Again, this research poster lists several sort of references that can be accessed online. The full research poster is accessible through the Renal Patient Support Group, the RPSG ResearchGate page, and so listeners are more than welcome to access the works through that way. And so this closes this audio podcast around this particular overview in and around this works on transplant neuroimmunobiology, the immune system, transplant rejection, and neuroendocrinology are retrospective, where the works has been put forward for the International Paediatric Transplantation Association, or IPTA 2023. Thank you for taking the time to listen in on this overview.